and welcome to this tutorial on the challenges of methodological variability and experimental design in EEG. During this presentation, I'm going to go through some of the methodological challenges that you'll likely face when you're going to be recording and analysing and interpreting your EEG data. So for those of you who worked in the field for a while, then some of what I'll talk about will be very familiar. But if you're just starting off in EEG research, then hopefully it'll provide you with some of the fundamentals that are important to think about. So at the end of the slides, you'll find a list of references which you can use to build on the topics that I'm going to go through today. So as with all forms of brain imaging, EEG presents its own set of challenges and pitfalls. You're going to have to manage through your choice of recruitment, of recording, and of analysis parameters. So as we don't have time here to go into detail about every single one of these, I'm going to focus on 4 key ones that we think especially important to think about when doing EEG research. So these are the definition of EEG frequency bands, selecting references, reporting of methods and magnitudes, and dealing with inter and intra-individual differences. So we'll take a look at each of these in turn, go through what some of the issues are, and then I'll put forward some suggestions for best practices about how you can try and manage them. So the use of frequency bands to analyse the frequency spectrum, so in other words, the method of splicing up the spectrum into bands of alpha, beta, theta, delta, and gamma, is one of the dominant analysis approaches that you're going to find in the EEG literature. So the method of data analysis has its origins in the 1930s and 1940s in a pre-computer era, so when there were limits on how you could analyse such complex spectral data. And the use of bands first emerged right at the start of EEG history with the discovery of alpha oscillations, and you can see here on the left an early recordings showing the classical effect of alpha blocking when a participant is performing a mental calculation. And this band-based approach has perpetuated through history, and in the middle you can see W. Gray Walter, and he's standing next to a device called the Walter analyzer that he built in the 1940s to divide up the EEG signal into a series of fixed frequency bands. So something that back then was even considered a suboptimal approach, but obviously necessary because of the technological limitations of the time. So these days we've got much greater computing power available to us compared to back then. So the picture on the right just shows the kind of machine that was used to analyze the EEG in the 1950s in MIT. And it's obviously rather different from what we know today. So, but despite the technological advancements that we've had, and on the right here you can see Moore's law as a reminder of the exponential level of growth that we've seen in computing power since the 1970s. The approach of slicing up the frequency spectrum into these 5 macro bands continues to persist today. So this is despite the fact that it results in a reduction in the temporal information that's available within the EEG data, and something that's meant to be one of the real assets of EEG over other imaging techniques such as fMRI. And so reporting increases and decreases in these 5 frequency bands still remains the norm for much of the EEG literature. And they're also used in the search for clinical markers such as the theta-beta ratio we just heard about, that has recently and somewhat controversially been approved by the FDA as a biomarker for ADHD. However, simply relying on combinations of increases and decreases in these 5 macro bands to explain all cognitive processes or disorders in the brain is probably unlikely to enable us to kind of achieve a more fine-grained view of what's actually going on. And this is why researchers are increasingly exploring other measures which don't rely on cutting up the spectrum into these bands, and some of which we've been hearing about in the Symposium. But there's another important issue here too, and that's in relation to the consistency or the inconsistency in the values that researchers are actually using to define the start and the ends of these frequency bands. So to explore this, we did a review of a set of 184 resting state EEG studies which compared clinical patients against healthy controls to see what kind of frequency bands windows people were using. And what we found was that across the studies there was pretty considerable inconsistency in the definitions that people were using. So if you look at the figure below, it shows the variation in the way that the different bands were defined across all the studies. The thick part of the bar shows the most common frequency band selected for use across all the studies, but typically this band was only used in about 30 to 50% of the studies, depending on which band it was. And what's particularly worrying is the thin line. So take a look at the thin line. It shows the variation in the frequency windows being used. So for example, if you look at delta, and the actual values you see in the table above, where min equals the minimum start value and max indicates the maximum end value for each band. And so for delta, it ranges from a minimum start value of 0 hertz all the way through to a maximum end value of 6 hertz. So that's quite far into territory that we would typically consider as being theta activity. Similarly, the borders between theta and alpha and between alpha and beta are pretty poorly defined. So where does one band end and the next one start? Well, if you took a look across all these 184 studies, it would be pretty difficult to say. So even if you take a look at the most common ranges used in the 30 to 50% of the studies, the values still don't match up. There's still gaps and overlaps in the numbers. So the implications of this is that what one publication means by, for example, delta or beta is not necessarily the same as what another publication means by the same terminology. So one researcher's beta might be different than others, but they're still both calling it beta in their results section. And so it makes it quite difficult to compare across studies because you don't know whether the differences that you find between the studies are due to differences in the frequency band definitions or because of something more meaningful. And although most of the studies we looked at did report what windows they used in the method section, so you can keep track of the differences that there are, an alternative solution is to use a more global standard for the definition of these bands, similar to what we currently use for, say, the 1020 system for electrode layout. So another thing to consider is whether you're reporting absolute or relative data. So for example, when we looked across the studies that analysed both types of data, they sometimes showed a different pattern of results for the absolute versus the relative. And so what's more, in the set of studies that we looked at, the relative findings were also often more consistent. And therefore, when you're deciding how to analyse your data, considering both absolute and relative approaches is something worth thinking about, and also has implications for when you're comparing across studies and data sets. And more generally, and based on the points I mentioned earlier about how the historical approach of slicing up the frequency spectrum results in a loss of the temporal information within the EEG, exploring alternative analysis approaches which don't require you to divide up the frequency spectrum in this way are definitely worth considering. So in the meantime, what could this global standard for the definition of frequency bands look like? Well, based on the data that we collected in our resting state review and taking into account the most commonly used bands and also the gaps and overlaps, it could look something like this. So delta would potentially be between 0.1 and 4 hertz, theta between 4 and 7.5 hertz, alpha between 7.5 and 12.5 hertz, beta would then be 12.5 to 30, and gamma would be 30 to 40 hertz. So the choice of frequency bands that you use in your study is only one of a whole number of methodological parameters that you need to think about when you're setting up your EEG study. So another fundamental one is the choice of reference that you're going to use. So as quoted here by Steve Luck, so if you don't fully understand referencing, you won't understand the signal that you're recording. However, the difficulty that you're going to face is that reference choice is still an unresolved dilemma in EEG research, and there's no best option. So there are a number of options you can choose from, so ranging from the nose and CZ through to linked mastoids and average reference, or even REST, which stands for Reference Electrode Standardization Technique, sometimes called the affinity reference. And it's a newer reference which was introduced in 2001. But each of these references has its own set of limitations. Currently, the two most popular ones, mastoids and the average reference, although there is increasing kind of traction behind the newer REST approach as people try and find alternatives to these more traditional reference methods in the search for an ideal reference. So what would be an ideal reference? Well, so it's one that is basically one that's electrically neutral. Otherwise, it can potentially lead to a distortion in your results. So for example, the choice of mastoids behind the ear is based on the assumption that the sites around the mastoid bones are free from activity of a neural source. However, we know that this assumption is actually incorrect because in truth, there's no single location in the body where the activity can be considered completely neutral. And therefore, there's always going to be some level of noise and signal distortion in your data. Similarly, an average reference will fall foul of the fact that you can't record from the underneath surface of the brain, and therefore the average is never going to add up to 0. And what this all means is that your choice of reference can potentially make a difference to the results that you will get from your study. So if you choose to use linked mastoids, for example, then you may potentially obtain a slightly different pattern of results compared to if you use average referencing or some other method. And what's more, your choice of reference is influenced by other experimental parameters. So for example, the density and the number of electrodes that you're using will have an impact on the average reference value. So in general, the average reference is recommended when you have a higher density of electrodes which cover the majority or as much of the scalp as possible. And all this is relevant regardless of whether you're going to be looking at the power spectrum when analysing, for example, resting state EEG data, or whether you're going to be looking at evoked potentials when participants are performing a task of some kind. So if we take a look at some data to see what the reference impact actually looks like. So here are two sets of results taken from power values, but there are also other studies which have looked at how the choice of reference influences other measures of kind of connectivity and coherence, complexity, as well as in setups which combine EEG and fMRI. And in the reference section at the end, there's a bunch of papers there which cover how references influence those other metrics. So on the left is a study investigating the effect of different references on an eyes closed resting state EEG data. And here you can see the way that the reference choice influences the absolute power values across the different frequency bands. So the graph in grey, so the results there show that there are differences in the amplitude values across the different references, with the left mastoid generally showing the greatest power values and the average referencing showing the lowest. And when you look at the spectral maps, you can see how this translates at the level of the whole head. And what you find is that because of the position of the mastoids in the head, it differentially influences the pattern of EEG activity that you see compared to the average reference. So for example, the mastoids are closer to the occipital region, and so they have a differential effect on the power of occipital electrodes compared to frontal electrodes. And this results in a shift in power towards the anterior regions compared to the rest and the average references. And similarly, because the left mastoid is closer to the left side of the brain, the strength of the left posterior area is decreased while the strength of the right anterior area is increased compared to the other references. And researchers have also looked at the effect of referencing on other parametrics, such as the alpha asymmetry measures, so shown on the graphs on the right. And again, they see that the choice of reference makes a difference to the pattern of results you get here, especially over the frontal regions, which are shown to the left sides of the graph. So if we now turn to ERPs, we can see that the choice of reference has an impact here too. So for example, on the left is a study which used a visual oddball paradigm with normal and rare targets and looked at the P300 response to these targets using different reference schemes. And what they found is that although the effect between the targets and the normal stimuli wasn't really affected by the choice of reference, the general amplitude of the P300 effect was influenced by the choice of reference. So in this case, it was the linked mastoid reference which resulted in the largest P300 effect during the task, followed by the rest, and then the average reference. And a similar pattern is found when looking at other ERP waveforms. So for example, in the middle is a task which investigated the feedback-related negativity, the FRN, which is a waveform associated with expectancy violations. And they found the same thing, that the comparison of win and loss trials wasn't so much affected by the choice of reference, but the overall amplitude was. So again, with the linked mastoids showing the greatest amplitude, followed by the average referencing, and then the rest. And finally, on the right is data from a study which explored the ERP response to emotional faces. So this study was motivated by the fact that in a review of the literature, they found that the choice of reference seemed to be having an impact on whether researchers reported a significant effect or not. So those who would use an average reference in their studies seemed to be more often reporting a significant effect of emotion on the N170, so an ERP component related to facial processing, compared to those who had used the mastoid referencing. And indeed, when they directly compared these two types of referencing in their study, they found a difference in the amplitude. This time, they found a more negative waveform for the average referencing compared to the mastoids over the posterior regions. And so what all this suggests is that the choice of reference doesn't necessarily influence task-relevant effects, but through its ability to shift the overall amplitude, then the choice of reference can potentially change the likelihood of you finding a significant effect in your data. So what are the implications of people choosing different references? Well, firstly, when studies are using different references, it can make it much more difficult to compare between them. So similar to with frequency bands, you don't know whether a difference between studies because of the choice of different references or because of something else. So what can you do as a researcher? Well, given the impact of a reference choice on the EEG, it's maybe worth considering whether it merits more than just mentioning briefly in the method section, or whether the potential implications of your reference choice should also be discussed maybe in the discussion and limitations section of your article. Similarly, including or making sure you include a justification as to why you chose a particular reference in your method section is something to think about, especially given there's no such thing as an ideal reference. And finally, it's also important to consider a researcher's choice of reference when you're comparing across studies or across data sets, because obviously it will have an impact. And as the search for, the optimal reference continues, so something that's probably not going to be solved anytime soon because it's been around since the earliest EEG recordings, working together with, your collaborators and co-workers and to be maybe more consistent in the choice of references that you're using can go some way to mitigating these challenges. So especially if you're working in the same research area or if you're working on the same clinical disorder. So referencing and the definition of frequency bands are two areas where there's considerable inconsistency in the choice of parameters used across studies, and so that obviously can have impacts on the results that you're going to get for your EEG study. The main consequence of this variability, as I said, is it makes it really difficult to compare across studies to find consistent patterns of effects or to look for meaningful biomarkers, for example, for particular groups of patients. But another thing that makes it quite difficult to compare across studies is the fact that people don't always report all the parameters that they're using. So given the complexity of analysing something like the power spectrum, there are many different parameter settings that you can choose and which potentially make a difference to your results. So if you change those parameters, then you potentially change your results. And so only by including all the necessary parameters in your method section can your readers really see what you actually did in your analysis. So, for example, when we looked across the group of resting state EEG studies, we found ones in there who included all the recommended parameters in relation to how they performed, for example, the Fourier transform in their analysis. But we also found studies that simply stated what software program they used without noting any of the specific parameters. And so as a minimum, this is the kind of information you need to be including when examining the power spectrum. But there are obviously other details that specifically relate to recording ERPs. So things like the electrode type, the material and montage and positioning, so the reference method that you're using, the recording duration and epoch length, what kind of artifact rejection or correction method you used, what filtering methods you were using, what algorithm you used for your Fourier transform and the different details of that, whether you were analysing absolute or relative data. So in our review, we found that actually 30% of the studies didn't specifically mention whether they were analysing absolute or relative data. So for those, we just had to rely on the assumption that it was absolute. But obviously, it's much better if it's actually mentioned in the method section. And also for eyes open and versus eyes closed, and the frequency band windows that you're using. So there are a number of publications which outline the best practices for what you should be reporting in your method section. So I put a couple of references at the end of the presentation, which are worth taking a look at. And one really helpful resource is an article by Keel and colleagues. And in this article at the end, there's an appendix which outlines a checklist for authors to go through when you're writing up your EEG study. So it's definitely worth taking a look at that paper. But the bottom line when you're writing out your method section is that you need to provide enough detail so that the reader can not only understand what you did, but also so that they can easily use the same parameters as you did. And so making it easier to compare across different studies. And also by including all the recommended details, It also means that if anyone wants to replicate your study, then they can easily do so. So another thing that's important to report in your article, and this time in your results section, is the magnitude of the effects that you're actually finding. So again, when we looked across the group of resting state EEG studies, we found that only about 40% of the studies actually provided kind of an easy to decipher graphical or numerical value for the magnitude of their results. So without this information, it was really difficult to work out how big an effect there was in the study. So in other words, by how much the alpha or the beta power actually increased or decreased between the different experimental groups. Instead, we had to simply rely on the reporting of the significance or the p-value. So for example, when we looked across the range of magnitudes that were reported for significant findings, we found a wide variety of values, so ranging from small magnitude increases or decreases around 10% through to much larger ones of around 60 to 70%. So in other words, some studies were reporting a significant result, but the magnitude of the effect was quite small, while other studies were reporting a significant result, but the magnitude was much larger. So in the former, the result statistically significant, but could potentially be less meaningful compared to the latter one with the larger magnitudes. And this also links to wider discussions that are going on around the best practices for kind of the way you explore and report your data significance. So now we'll turn to the final challenge that I'm going to run through today, and this is individual differences. So this obviously comes in two forms, so the differences between people and the differences within people across different moments of time. So in general, inter-individual differences are usually bigger, but both are still important to think about. So however, individual differences are often rather neglected in EEG studies. This is because there's usually a need to average across the whole group of subjects to ensure that you've got good enough signal-to-noise ratio in your data. In addition, the data is usually only collected from a relatively small number of people, and each person is usually only tested once. So let's start with intra-individual differences. So what kind of intra-individual differences might you need to consider? So when a participant comes into your lab to take part in a study, you'll likely get them fill in a questionnaire. So one thing to think about is what kind of information do you need to be asking them? So obviously you need to ask standard information like age and gender and left-handed, right-handed, that kind of thing. But what else? Well, there are a couple of things you might not immediately think about asking, but could be influencing the kind of data that you're collecting. So for example, there are various substances which are known to potentially have an impact on the EEG. So we'll take a look firstly at a couple of examples, for example, of caffeine on the EEG. So on the left is a study which looked at the effects of caffeine on the power spectrum using a randomized, double-blind, placebo-controlled study. What they found was that the ingestion of caffeine resulted in a decrease in spectral power when people had their eyes closed in an eyes closed resting state setup. And this decrease was mainly observed over the alpha band, suggesting an increase in arousal. However, the effects of the substances aren't always completely straightforward. So, for example, the graph on the right also looked at the effect of caffeine, but took into account how sleep deprived the participants were and also controlled for withdrawal effects more strictly. And what they found was that there was an interaction effect in the data. So when participants were well rested, they didn't really actually show any significant effect of caffeine. In contrast, people who were sleep restricted showed a significant effect of caffeine, this time on their beta power over posterior regions in the form of an increase in power compared to the placebo condition. So in other words, the effects of caffeine, you can't just consider them in necessarily in isolation. You should be considering them in the context of other situational factors as well, like how tired and sleep deprived your participants, and also whether they're currently in a state of caffeine withdrawal or not. So similarly, other substances have been shown to have an effect on EEG. So for example, studies have explored, on the left here, the effect of glucose on the power spectrum and shown increases in alpha amplitude after taking in a glucose-based drink compared to a period of fasting. And then on the right is other studies which have looked at what's the effect of having an alcoholic drink on the EEG. And they looked particularly at patterns of connectivity through measures of coherence across the brain and found changes in the alpha, theta, and beta bands that kind of reflected the changes in functional connectivity after people had drunk some alcohol. But it's worth noting that the measuring the effects of substances on the EEG isn't necessarily always straightforward. So there's lots of confounding factors, and also the literature could do with a greater number of studies, also with larger N's, looking at the impact of substances like these on the EEG. But what it potentially means is that if one of your participants has drunk an energy drink just before they came to your testing session, then potentially you could be getting a different set of results compared to if they just had, say, a glass of water. And if they skip breakfast, then you might see a different pattern to if they'd just eaten a chocolate bar. So what can you do to help manage intra-individual differences in your data? Well, there's a couple of things you can think about. Firstly, you can ask people to control what they eat or drink before they come to the session. But it can be complex to do this because if you ask someone to refrain from drinking coffee before the session, it can potentially have a differential impact on people who are regular coffee drinkers compared to those who drink coffee. Secondly, you can collect more than one data set per person. So this means that you aren't relying on a single session of data to reflect the individual, but you can use multi-session data to take into account any session-specific differences. And thirdly, you need to collect sufficient metadata so you can take into account these potential differences in your data. So Sapien Labs has a session form called a state of mind form, allows you to collect this kind of data. And the link here takes you to the form that you can download for use in your studies. So what about inter-individual differences? So the variation that arises from the fact that everyone's brain is developed slightly differently depending on their genetics, on their developmental trajectories and their life experience. The one inter-individual difference that's commonly considered in EEG studies is the age of your participants. So this is because obviously the EEG spectrum is known to evolve with age, and so by controlling for age or by properly age matching your participants over the experimental groups, you can take into account any unwanted effects of age on your data. So for example, the amplitude of the P300 is shown to evolve over the lifespan, as shown here on the left. And also the power of the different frequency bands varying according to age and also gender. So something that's been demonstrated in various studies, including this one, which is a large-scale study conducted with the Muse headband shown on the right. But inter-individual differences go much further than just the age of your participants. So there are many other factors in relation to a person's life experience or developmental trajectory which could influence their EEG. And simply collecting data from a small number of participants, with having a small n in your study, doesn't necessarily allow you to either explore or control for these differences. So for example, in our review of resting state studies, we found that the median number of participants in the study was 60. So if you look at this value in proportion to the global population of about 7.7 billion, then you're only considering data from a tiny fraction of the global population. And only by significantly increasing the N and recruiting participants who kind of reflect the more broad diversity of the global population, can you start to address the issue and also the opportunity of inter-individual differences. So there's one simple way to increase your N, and that's through collaboration or by establishing kind of multi-site consortiums where different labs collect EEG data using the same experimental setup. So something that obviously requires a standardization of methodologies. So by collaborating, you can aim for not just tens of people in your study, but you can aim for hundreds or thousands or even hundreds of thousands. So you search for key brain effects or clinical biomarkers. And means you can truly take into account the diversity of brain function and also dysfunction. And this obviously requires you to collect standardized demographic information about your participants, and it's something you can do. So we have a subject form group, so the link here will take you to the form which you can download for use in your own studies. So in summary, we've looked at a couple of methodological challenges that you'll likely face when conducting EEG studies. I hope you found the tutorial session useful. So if you want to do any further reading then on the topics I've mentioned, I've included a list of references that you can take a look at when you get a copy of the presentation. So hopefully that will build on what I've already said.